Okay, this is part of a series of videos to help you with suggestions that may help enhance the report. You do not need to do all of these things, and likely you should do much more than what's written, but these are just suggestions to help guide you to things that you should likely consider when writing your lab report. So it's not an exclusive list, it's not an exhaustive list, but it's certainly things you should look at. If you don't cover them, that is fine as long as you cover something. So they're a series of three. Each one is dedicated towards one of the Fox laboratories. So this one will be 8.4, looking at the acid-base water buffer, basically aspect of this. And then the other two will be 1.3 and 2.1. So in general, here's some things you should consider. Certainly look at JLR 13 and double check it. That's the checklist that tells you what you need to cover in your lab report, and make sure legitimately you've looked at those things and you can check them off. Also look at the common errors. There's a video and handout available for that, and make sure you have avoided those. So anything having to do with 8.4, here are some of the things you ought to consider. You'll see some of these again in each specific lab. But one of the things you should look at is what is pH. You should probably then look at what's an acid and a base. You can use different definitions. It's fine. Whatever suits your fancy is fine. You should probably, for all of them, mention what a buffer is. Certainly some obviously have a much greater role in terms of the buffer, like the experiment where we used buffer. But even the experiments we're looking at for hyperventilation or the blowing through the straw after exercise for the time to color change, important aspect of it is why doesn't pH in the blood change very much? So you should talk about acid-base balance and that control. You should talk a little bit maybe about acidosis and alkalosis, the pH measurements. So what is considered acidosis? 7.35 below. What is considered alkalosis? 7.45 below. What are the complications with that? How does the respiration help control blood pH? How do the kidneys metabolically control pH? Certainly, you probably should look at how CO2 acts as an acid in your blood. And the way it works is this equation. Certainly, most of you will probably consider this equation as part of your lab report if you're doing one of the labs associated with this. So as an example, this helps to show how CO2 acts as an acid in your blood. So as we gain more CO2, it's going to shift this to the right. So it's going to go that way. And because of the way this equation works, notice it's bidirectional, it's got double arrows on both sides, we'll make more H plus. Okay, so that's the hydrogen ion. Remember, pH is a measure of the free hydrogen ion concentration. So you probably should understand this equation and likely explain it for your particular um, uh, laboratory. It also explains, as an example, let me erase everything, how hyperventilation increases blood pH, lowers the hydrogen ion concentration. So as you hyperventilate, you're getting rid of CO2. So I'm going to put a little arrow up to say you're getting rid of CO2. And as we get rid of CO2, that's going to take this equation and drive it in this direction. So it's going to go from right to left, because as CO2 gets blown off from your lungs into the atmosphere, it's going to make this side of the equation lower, which just by mass action is going to drive the equation to the left. So for the hyperventilation one, that's what happens. If you want to explain how it works in the blown through the straw one, right? Before exercise, you have normal CO2, and it makes a normal amount of H plus. After exercise, because of the increase in metabolism, your CO2 goes up. Since your CO2 goes up, that's going to drive this equation this time to the right, because I've got more CO2 on this side of the equation, so there's more here, and then there's more here. The law of mass action again is going to make this equation go this way. When it goes this way, the hydrogen ion concentration is going to increase. It's going to go up. And if we increase the H+, we're going to decrease in pH. And it's that decrease in pH that changes the color in the time to color change for the pink to clear before and after exercise. So in all of these situations, that's an important equation. And you'll see it in all of them, but just kind of that's what we'll see. So let's go ahead and look at each individual lab in a little more detail as well. So the first lab would be the one dealing with acid-base water buffer. So again, you should check out FOX and 8.4a. What also might help is the lecture S1 PX2. So that's the basic chemistry in the modules as well. It kind of explains a little bit about acid and base, some of that information you might be able to use in your report. Some of the things you should specifically say in this one is, since we're directly measuring pH, right, with a pH meter, what is pH and how do we measure it? You should talk about what is an ionic bond. Remember, we said all ionic bonds dissociate in water, so they break apart. And the things that are held together by ionic bonds would be salts, acids, and bases. And so how does that work, and how does that affect when we add the NaOH or the HCl into the water, the buffer? You should mention probably how acid is different than a base. Since we're using a buffer, you should talk about how buffers work. Eventually, that probably should relate to the 3 primary buffers that humans have. That will also go into acid-base control. Again, you probably should talk about the standards for acidosis and alkalosis, those medical conditions where the pH is not in the normal range. You should, maybe mention the complications that you have with acidosis and alkalosis if your pH gets too high or too low. Certainly mentioning how the respiration and the kidneys help to control blood pH. You should talk a little bit about how CO2 acts as an acid in your blood, and also how metabolic acids affect blood pH. And maybe even on the other side, is it possible to have a base excess in our body? It certainly is. And maybe what are the conditions that cause that? That equation we mentioned before, right, is important. And for your data, you ought to consider separate tables. Since most of the time we're not going to be comparing acid and base, we're going to look at what happened to the acid in the water and the buffer. So that should be one table, right? So one table for acid and water and buffer, and the other table for base in water and buffer. And that way we can see the relative changes with 1 drop and 3 drops, and kind of looking at that. But again, there's a lot of different ways to do that effectively, and that's just one of them. So that's the acid-base buffer one. All right, now we'll look at the exercise and time to color mentioned. That's in FOX 8.4b. Other information that might help would be section 1, PX2. That's the basic chemistry lecture in the modules. That explains a little bit about acids and bases. What is pH and how we measure it would be an important variable to talk about, since it's the pH change that we're actually measuring with the phenolphthalein. What happens to blood pH when we exercise, and why it doesn't really change. So when you exercise, blood pH doesn't change much. And that's kind of an important idea, and it's because of what we call respiratory compensation. That respiratory compensation has to do with this idea of hyperpnea. Okay, so when you look at this word hyperpnea, hyper, increased, pnea refers to breathing, like apnea, apnea would be someone who doesn't breathe at all. A was without, pnea means breathing. So hyperpnea means increased breathing. Well, if we look and say, oh, there's also a similar word if you translate it, hyper, increased, ventilation, breathing. has a totally different meaning. So you should make sure you understand the difference between the two. Hyperventilation and hyperpnea. You probably should discuss what a buffer is and what the three primary buffers in the human body are. There's a bicarbonate buffer, which has to do with this equation right here, down here. This is bicarbonate down here. But you also have a protein and a phosphate buffer. You should talk about, maybe acid-base control. What are the measurements for acidosis and alkalosis? At what pH measurements do they come in? Maybe, again, if you're looking for more information to talk about, what are the complications of acidosis and alkalosis, and then how the kidneys and your respiration helps to control that thing. Probably should talk about how CO2 acts as an acid in your blood. And that becomes very important because when CO2 is in the blood, when you exercise, it's going to increase the hydrogen ion concentration and how it does that, and then how blowing off the CO2 in respiration normalizes that. And so as long as your respiration matches your metabolism, then you should be fine. This one for the data is pretty easy. When you're looking at your data, consider using a single table or figure. You don't need much more than that. And I would probably, if it was me, just make a pre-exercise and post-exercise table. So again, if it was me, I would probably just consider making a pre-exercise and post-exercise table with the time to color change. Again, for your tables and figures, make sure they're all complete the way they should be. And you can look at that in the other information that is posted. Okay, and the last part of this would be 8.4c, which would be the effect of hyperventilation on respiratory rate. So again, obviously looking at FOX 8.4c, the basic chemistry, section 1, PX2, and then things you want to look at was what's normal respiratory rate? What is it? What controls it? Right? Again, since we're looking at hyperventilation, make sure you understand the difference between hyperventilation and hyperpnea. What happens to blood pH when we exercise and why it doesn't normally change? That's an important thing to look at. We are not exercising per se, but some of the explanation on why the data looks the way it is because we hyperventilate so vigorously. It's actually like doing exercise. What is pH and how we measure it is important. Again, the respiratory compensation, the three human primary buffers we'll see in the body, important. What's acid-base balance control? So how we control acid-base balance, how we, again, determine acidosis, alkalosis by primarily pH measurements, what are the complications for those diseases, how respiration and your kidneys metabolically help control pH. This one's important, how CO2 acts as an acid in your blood. As you hyperventilate, you get rid of this CO2. That drives this equation that way. And so you decrease the hydrogen ion concentration there. And since there's less hydrogen concentration, you have less drive to ventilation. Your breathing rate should slow. To show the data, it's pretty simple. Just use a single table or figure to show the hyperventilation, both pre and post, when you're looking at the respiratory rate. So just show the respiratory rate, pre-hyperventilation and then post-hyperventilation. So that should be a pretty good idea of the types of things you need to do on this and you'd want to look at when preparing any of the sections from FOX 8.4.